So the next speaker is Dr. Doug Munoz. He's a professor in the Center for Neurosciences Studies and Departments of Biomedical and Molecular Sciences, Psychology, and Medicine at Queen's University. He currently holds a Tier 1 Canada Research Chair and is co-lead of the ONDRI study. So welcome, Doug.
Thank you. Good morning, everybody. And it's a great opportunity for me to be able to talk to you today and to continue on this theme that Mario introduced about the search for biomarkers and the search for early biomarkers of disease so that we can hopefully intercept the disease trajectory earlier and ultimately be able to slow it down or stop it. And I'm going to focus my presentation this morning on the hunt for a blood test to diagnose neurodegenerative diseases, but I'll also show a little bit of data of other kind of biomarkers of neurodegenerative disease and the work that we're doing within ONDRI to hunt for those biomarkers. And so Mario nicely introduced the foundational study of ONDRI. And just cleaning up my desktop here. And the different disease cohorts that went in: cerebrovascular disease, frontotemporal dementia, Parkinson's disease, Alzheimer's disease, mild cognitive impairment, and amyotrophic lateral sclerosis. 520 patients, and they were followed longitudinally. And as Mario introduced, there were a number of important assessment programs, platforms, a full clinical workup for all the patients, a blood test, a blood draw that was taken so that we could do genetics and genomics. And I'm going to come back to those blood samples later in my presentation. And then assessments of a number of behaviors, neuropsychological performance, gait and balance, eye tracking, and then imaging, brain imaging, and retinal imaging. And all of these data were collected from these patients. So we had 520 participants enrolled, and really the hunt was on for early biomarkers across all of these platforms and trying to identify where we could have the most reliable measures of these different forms of disease that Mario introduced. So I'm going to go way down in the brain, down to the molecules, to talk a little bit about the start of the disease process. So this is a schematic from deep in the brain, and this red membrane here is to be representing the surface membrane of a brain cell. And so in dark down here, this is inside the brain cell, and up here is outside the brain cell. And in all the cells we have in our brain, they're very complicated with these different proteins that might stick through the membrane, connecting from the inside to the outside. And this highlights just one of these proteins called APP that has this transmembrane domain here. And there are enzymes that are going to come along and cleave this protein regularly through life and break it into small components that are called the oligomers. And then this is now circulating in the cerebrospinal fluid in your brain. And when you go to sleep, these kind of molecules, which are like small toxins almost, they're waste products, get flushed out of your brain. But in some individuals, and for some reasons that we don't know, these oligomers, which are sticky, can start to clump together. And they make these fibrils, and then these clumps get bigger and bigger, and eventually form these large plaques. And many of you have probably heard about plaques in Alzheimer's disease. And back 30 years ago or so, when I was a graduate student, this was believed to be the cause of Alzheimer's disease. However, we learned a lot more about the science along the way. And so this is this same pathway of the oligomers forming plaques. But these oligomers also circulate through the cerebrospinal fluid and can start to attack other brain cells. and start to damage the synapses that are on the outer processes of these other brain cells. And so this schematic now shows how this might look if we had a healthy brain cell shown more in red here, and an unhealthy brain cell, a diseased neuron, because various factors have made this neuron, this brain cell, prone to dying, including the attack of those oligomers. And now if we look inside one of these processes of this diseased neuron, and we compare it to a process of the healthy neuron, we start to see problems. So in the healthy neuron, there's a network of what are called these microtubules that are used to move various molecules and vesicles up and down from the cell. So material needs to move from the center of the cell out to the periphery and vice versa. And this network of microtubules this is like the highway that those molecules are moving on. And as these neurons get ill, these highways, these microtubules start to break down, and the proteins here start to be released, including tau. And the tau here, shown in blue, can become hyperphosphorylated, so that now it starts to clump together, kind of like the oligomers did outside the cell. Now the tau is clumping together inside the cell to make a tangle that's going to make this cell start to die. Here's another molecule in the brain called neurofilament light. This is a brain cell. It has a process which is called an axon, which is going out to create synapses, and these are the connections to new brain cells. So this axon is how the information's being propagated. And if we look inside the axon, we can see these other molecules called neurofilaments, providing this long scaffolding structure in the axon. And this neurofilament light, if the axon is damaged in some kind of way, this neurofilament light can start to break down and then circulate within the cerebrospinal fluid at some concentration. And evidence of this neurofilament light means that there are sick and dying brain cells. And what's interesting is some of these markers can manage to get out into the blood. And I'll come back to that later. And so neurofilament light is another one of these potential markers that appears in multiple neurological diseases, including the ones we study in ONDRI, like Alzheimer's and dementia, stroke, amyotrophic lateral sclerosis, and Parkinson's disease. And so in Alzheimer's disease, some of these markers like A-beta oligomers go down. As I mentioned, tau levels in the CSF go up. Neurofilament light increases in the CSF. So these are potential biomarkers to tell us who might have Alzheimer's disease or be leading to getting Alzheimer's disease. And I literally cut this slide from Dr. Maselli's talk 20 minutes ago and inserted it in my own because I didn't have this link. And he talked to you about these proteinopathies. And so I showed you beta amyloid and tau, which are the proteinopathies for Alzheimer's disease. And in the other neurodegenerative diseases, similar problems are happening, but it's a different protein that's having problems. So for example, in Parkinson's disease, as Dr. Masella said, it was the synucleinopathies. It's TDP43 within frontal temporal lobar dementia and ALS. And there are other tauopathies in frontal temporal dementia as well. And so there are a number of different proteins that are going awry, the coding of those proteins in the brain, and these are all producing potential biomarkers in the cerebrospinal fluid if we can figure out how to measure them. So this is a wonderful schematic diagram now that's about 10 years old that sort of shows the progression towards Alzheimer's disease. So on the horizontal axis, this is time. And I put two markings here. The time when an individual would go from being preclinical to a neurologist or geriatrician might give them a diagnosis of mild cognitive impairment. And then this second line would be when the clinician would switch the diagnosis now to Alzheimer's or dementia. And if we look at the pattern of changes in the brain over time, before and after these diagnostic signposts, there's important things I want to show you here. So first of all, when we look at the molecules in the cerebrospinal fluid that are going abnormal, the beta oligomers, tau, those molecules are changing and their levels are increasing well in advance of any clinical diagnosis. This, we believe, is the earliest part of the disease process. And it could be happening 20 or 30 years before the clinical diagnosis. And those proteins that are accumulating and those abnormalities are starting to attack neurons. They're leading to sick brain cells and synapses that are disintegrating. And so synapses are starting to be lost here. Still, before the earliest clinical diagnosis takes place. And the loss of these synapses is going to have an effect on the networks in the brain so that the functional connectivity will start to change. Again, the earliest changes happening before any clinical diagnosis. And finally, all of those changes will lead now to behavioral changes where the patient or the individual might have learning or memory deficits or executive function deficits. And that's what's allowing the clinicians to make these diagnoses. But by the time the clinicians make these diagnoses, a tremendous amount of brain pathology has taken place. And so wouldn't it be great if we could go back here and find the earliest biomarkers in the cerebrospinal fluid or the blood so that we could alter the trajectory of these disease processes? That's really the goal of ONDRI and all the neuroscientists working on neurodegenerative diseases. So these biomarkers we can get from the cerebrospinal fluid are specific, they're sensitive, they're measurable, and we can see how those molecules will change to therapy. A huge problem is in ONDRI, we were unable to acquire cerebrospinal fluid from our patient volunteers. This is an invasive procedure. It requires a spinal tap that can make the patient quite uncomfortable. And all of these biomarkers are in the cerebrospinal fluid, which circulates around the spinal cord and around the brain. And that's a unique fluid area in the body. And the reason is because we have a blood-brain barrier. And that's represented in this figure. So the top part here would represent a blood vessel coming from the heart and lungs. So it's bringing all the nutrients to the brain, oxygen, sugar, assorted other molecules that need to get into the brain down here where the cerebrospinal fluid is. But there's a barrier of cells here, a natural barrier called the blood-brain barrier, and it's not going to let anything in the blood come across into the brain. The brain needs to be well regulated. So only certain molecules can get across this barrier. And the result is that nutrients can flow one way across this barrier from the blood into the brain to nourish the brain cells, and waste material from the brain flows the other way out to the blood to be taken away. Now, these little red circles I put in down here are to represent one of these molecules resulting from one of these proteinopathies. Let's say this is elevated levels of tau in the cerebrospinal fluid or neurofilament light. And in ONDRI, we were unable to get a measure of cerebrospinal fluid. But some of these small molecules can get across this barrier and are therefore present also in the blood, unfortunately at much lower concentrations, meaning it's much harder to find them. So when we started ONDRI in 2013, there was no technology to really get after these biomarkers in the blood. But in the last 10 years, there's been a revolution in the technologies that are present that allow us to look for tiny amounts of these small molecules in the blood. And one of these technologies is SIMOA, single molecule array. And this just represents from 2001, when this technique was first discovered, to now the explosion in research publications trying to develop this technology to look for biomarkers in the blood for neurodegenerative diseases. And now it's a simple machine. We have an array here that gets put in the machine. It's ultra sensitive, and it will read the blood of patients and tell us if these tiny molecules are in the blood. And so remember I told you we had a blood sample taken to do the genetics in ONDRI way back at the beginning. Now all of this technology has emerged, and we have the opportunity now to do a molecular assessment of what's in the blood of the ONDRI participants. And we're looking for blood-based biomarkers here. So we collected the blood from the ONDRI participants in Ontario. Rather than try to create the new technology here, we took advantage of a collaborator of ours, Professor Henrik Zetterberg in Sweden, who's one of the world leaders at trying to find these blood-based biomarkers for neurodegenerative disease. He now has plasma samples in his lab from the initial ONDRI study, and he's looking for a number of molecular biomarkers in the blood of those ONDRI participants, including some of them that I talked to you about today. Now let me just switch gears in the last 2 minutes here, and I'll show you the kind of biomarkers I study. So those proteinopathies evolved in the brain, neurons, brain cells got sick, brain cells started to die. Brain cells normally make up these vast connections of networks all over the brain, and these networks are critical for controlling our behavior, our thoughts, our cognitive functions. But when the brain cells get injured and die, these circuits are going to be affected, and they're going to become dysfunctional, and it's going to lead to altered behavior. And that's what happens at the earliest stages in Alzheimer's disease when people start to have memory deficits. It's because the memory circuits here are becoming affected. I happen to study eye movements as part of ONDRI. This just circumnavigation of the brain shows all the brain areas involved in controlling eye movements. This slide shows the insane set of circuits of all the brain areas connected to control eye movements and blinks and pupil responses. So I'm an expert in this network. And I just want to show you one biomarker that we have from the ONDRI participants by measuring the size of their pupil while they were watching TV. And we were changing the channels. So this is to represent time. And every one of these little blocks is when we showed the patient a different channel. And at this red line, we went from where the screen was relatively dark to where it was much lighter. So the extreme of this is when you're in a dark room and you walk outside in the sun, it's really bright. One of the first things that has to happen is your pupil constricts. And you compare that when you're in bright light and then go into darkness. One of the first things that has to happen is your pupil has to dilate. And so we set up these changes in luminance in our eye tracking experiment. And I just want to show you one example here where I'm plotting in time the change in pupil size. And this is from a group of 100 control subjects showing when they went through one of these transitions from dim to bright, the pupil constricted. And when they went through one of these transitions that went from bright to dim, the pupil dilated. And when we showed this same experiment to patients with Alzheimer's disease, yes, the pupil constricted on the bright clips and it dilated on the dim clips, but there was a dramatic difference in the pupil response compared to the control subjects. The green line here shows the responses we got from Parkinson's patients. They were more similar to the elderly control participants, but again, dramatically different from Alzheimer's, reflecting the difference in the proteinopathy in these two different diseases. And so I won't go on any further showing you any more of this pupil disease, but just showing you how we're measuring the pupils of the patients and how they're dramatically different. And that's being driven by these proteins attacking these circuits in the brain that control these behaviors. And so I think I'll stop at this time and take any questions if there are any.